How to use CoreFlow. CoreFlow is the data management interface for analysis, data analysis and statistical analysis in molecular biology labs. First of all, we have to decide of a goal of a type of data analysis. I picked for now the arginine to proline conversion, and basically we want to estimate what is the conversion rate of the arginine to proline. I will explain about the SILAC and the context, the biological and measurement context a bit later. For now, I will show how to use CoreFlow to create a new analysis or an analysis task. So as you can see here, the most important highlighted item is this one. It's data analysis. I will click and I will create one example related to mass spectrometry and, as I said, to SILAC experiments, and more precisely arginine to proline conversion. This shows you also how the analysis or all the tasks are organized. They are collected together in a thread. The threads together can be collected, set under a certain project. For example, in this case, SILAC experiment, and multiple projects can be collected together under an owner, or they have an ownership. In my case, we already have this example of arginine to proline conversion, which is a copy of the previous one. So for now, I'm going to delete the old one. As you can see, it disappeared, so I no longer need it. I can do a refresh, and as you see, we end up with less projects, less owners. For this example, I'm going to still copy one of the tasks. So this is the existing example, estimation from experimental data. I'm going to copy and discuss just a bit about the content. This is a repeat of the placement of the task in the ownership, the project, and the thread. The most important fields are those ones. The one that has the analysis summary in wiki format. I'm going to click quickly and you'll see that this is the description of the task in a textual form, and this is the in a more nicer presentation. We don't need to discuss this right now. Usually the description of a new task appears at the end. Another area is data preprocessing. Because all the data is stored into the database, we'll have to use a certain language, structured query language, which is exemplified here. This is the language the database understands very well, and it's easy to write and to run this. This is a playground with a colored syntax, and I'm going to add a line, a command here, and I'm going to limit the number of results from a certain table, this table for example, to just 100. I'm going to run the script, and this is the result. So we'll see 100 rows in this case. This was an exemplification of the SQL query language. If you want to know more about the syntax and the grammar of this language, you can click on this online documentation. Coming back, we described so far the two areas or playgrounds, one for the documentation and one for data pre-processing from the database. And now I'm going to click on another one. The third one, in this case, is a piece of a script written in R that does all the data processing that we have seen before. As usual, you can run this script and obtain some results. I'm going to run it here. It says that is waiting for the results and cleared this bottom portion. And now the results are obtained in terms of some files. This is a PDF file. And I'm going to click and save it so that I can watch what is inside. We'll come back to the content of the script. It's suffice to say that it generates a number of graphs from which we can obtain experimental conversion rates of the arginine to proline conversion, for example, as compared to conversion to other amino acids. And besides this, it can give you the arginine to proline error, relative error. In this case, for example, there are many cases when we have around 10% relative error and fewer for much higher error. We can have cases up to 66% in relative error. This just briefly explains how all the system works. Once again, I'm going to close the tabs associated with this data analysis and come back to the CoreFlow. As I mentioned before, all the data is in the database. The data that has been used for the example, it's obviously in one of the tables. Here is the table. This is the table name. And if I click, I can get also the content of the table and the structure. Here's the structure with each column. So column has many values and the table has many rows. You can see what type of those values are. Could be a floating point numeric, or could be character, and so forth. You can sort based on a certain column. If you click on the column, it switches from descending to ascending order. And if you click on one of the columns in the structure, you can see what are all the distinct values that it has and in how many rows it has those particular cases. For example, here we have 6 cases, 6 rows with a charge of value 7. This concludes our small introduction to the CoreFlow. We'll go back to the details of what is arginine to proline conversion and why is it important for mass spectrometry next time. Thank you and goodbye.